So hi everyone, welcome to lab 3 of STAT 115 and 215. I'm Gigi Ken, you can call me Scarlett. I'm going to be your TA for homework 2. I have 2 announcements to make before we get into our lab materials. So first, homework 2 is already out and it's due February 21st, which is one week, like next Sunday, 2 weeks from now, by midnight. And everyone should already have a Canada account to proceed with your little homework. So here's some outline for our today's material. Today we're going to mostly focus on the first part of homework 2. And the next week, we're going to talk about machine learning and modeling accuracy comparison, which is the second part of the homework 2. So today we're going to cover PCA, hierarchical clustering, batch removal, DSIG2, K-means, David, and GC. These are the packages you will need to install to run this R Markdown file. So you can use either the install the packages or the BioC manager install to install this. So first, we're going to talk about the PCA. So often it's not helpful or informative to only look at all the variables in the data set for correlation or covariance. A preferable approach is to derive new variables from the original variable that preserves most of the information given by their variances. Principal component analysis is a widely used and popular statistical method for reducing data with many dimensions. By projecting the data with fewer dimensions, use the linear combination of the variables known as the PCs. The new projected variables are uncorrelated with each other and are ordered so that the first few components retain most of the variation present in the original variables. Thus, PCA is also useful in situations where the independent variables are correlated with each other and can be employed in exploratory data analysis or for making predictive models. So the PCA, the PCs are like the linear combination of variables, but it's not the best ones, just the ones that's describe the largest variation. In our case of studying the gene expression, we can use we can reduce our dimensions from very high dimensional to a lower dimensional matrix. So here's a little example of showing how the PCA works. So first, we're abandoning these two axes, which originally defines these dots right here. We take this new two dimensions. So first, we can see the PCA1 is the axis where the dots can project to and have the largest variance. And there are two observations for a second PC. The first one is we can see the second PC is if the dots project to the second PC is less variable than the first one, and the second one is we can see the PC2 is orthogonal to PC1. So that means PC2 is uncorrelated to PC1. So here is just a little example of the PCA it's a PCA analysis. So we're using one of the data set from online database, and it contains 23 individuals with 10 variables. If you're using a local directory file, data set or something, you can use the readout CVS and the readout table to read your data. Here, I'm just using this because it's just online data. And we're using the PRCOP to do the PCA. Here, I'm just doing some of this calculation to make my plots in a better sense. So from this graph, we can see that our first PC already covers 41.2 variation, and the second one covers 18.4, third one covers 12.4% of the variation, and so far so forth. So we can confidently say that the first PC already covers most of the variation within the data set. So we don't need to we don't really need to care about the rest of them. Maybe the first one, but not so further after the fifth one. So we already decreased our dimension from the 10 to maybe 3 or 4. Yeah, and this is how PCA does the dimension reduction. And if we assign our data points to 2 groups, so here I'm assigning the ones that can run 100 meters less than or greater than 11 seconds into the fast and low. We can see, like, we can quickly do a plot on the first PC and second PC, and we can see there is already a separation between the fast and slow. So the fast is on the left-hand side and the slow is on the right-hand side. So this is a very preliminary clustering method. So you're only based on the first 2 PC to see whether there is a clustering within like the categories that you assigned. And there are some more function that you can use to get some more information. So first one is the PCA result, which is a list that holds the mix of data types. You can access individual list elements with the $sign, with something like this. And if you want to know more of what is contained in this PCA result, you can just Google the PCA result. You can see all these documents. And there are a lot of our functions that's use list to return multiple things. So for all of these objects, we just use the $sign to get the individual list elements out. And actually, there are 2 ways of doing the PCA. The first one is the PR cop, and there's another function is the PRI cop. So this 2 uses different method to do the PCA. One of them using the eigenvalue eigenvector. The other one uses the singular decomposition. So if you want to know more of it, you can just Google them. There's a pretty good documents saying that describing the difference between them. And I'm not going to go really in detail for this one. Yeah, so the PC, so there can be as many as the PCs as your original variables. Each of the PC is going to be a linear combination of your original variables, and the PCs among the PCs, they're all orthogonal to each other, so they're uncorrelated. And now we're going to talk about K-means. So this is an unsupervised method for clustering, and so that requires you specify the number of clusters in advance. So clustering actually is a broad set of techniques for finding subgroups of observations within a data set. When we cluster observations, we want the observation in the same group to be similar and observation in the different groups to be dissimilar. So because there isn't a response variable, this is an unsupervised method, which implies that it seeks to find the relationship between the observations without being trained by response variable. So actually clustering allows us to identify which observations are alike and potentially categorize them. So K-means clustering is the simplest and the most commonly used clustering method for splitting a data into a set of K groups. So for data preparation, the rows are observations and the columns are variables. So in our case, the rows is individuals and the columns are the genes. And any missing value in this data should be removed or estimated. So if there's NA, just use NA to omit, or you can just replace this NA with some number. And the data must be standardized to make variable comparable. So we want the standardization to, we want the standardized data to have a mean around 0 and standard deviation of 1. So here is a cartoon to showing k-means with 3 centers. So we can so firstly we pick 3 random data from the data set, and we calculate the mean for the data points assigned to that cluster, and then we took the mean as the center and redefine the clusters and take the mean again to reassign the center. So after a couple of the iterations, we're going to get to a local minimum for within cluster distance. So this will not provide like the best separation, whereas only going to provide a local optimal. So for some of these data, there might be like the method might trap into a local minimum, and that might not be a good choice to do k-means. And how should we I'm going to talk about how should we choose for the k in a later session. Here, just showing you how to see the distance between samples. So we can use this getDist function to get the samples. We first need to scale and omit the data, as I said, omit the NNAs, as I said, and then we use the getDist to do to find the distance between each data points. And the default distance in R is Euclidean, so we're calculating the Euclidean distance. And we can see a visualization like this. So for some of them, maybe the red ones, these data points are further away from each other, and for some of them they're closer to each other, and this basically shows us how so this ones that's closer to each other is going to be clustered together, while this ones that's further away might not be the same cluster. And there's another way, another visualization you can use, which you can just delineate this boundary here, so you can have a better visualization. Yeah, just an option for you guys for a better way to present your data. And as I said, you can have different centers for different data sets. Here I'm showing you not only the 2 clusters, I'm showing you 3, 4, and 5. So you can see they can do a pretty good separation. But like for here, maybe 5, if you have 5 clusters, these are not too separable, which is not ideal. Yeah, so yeah, you can see like whether the k is good from this plot, or you can do something like this, where you plot the iteration, the center numbers with the within cluster distance. So we can see here the distance, the within cluster distance decreases as you increase your number of centers you pick. So let's say if we have you only have 10 data sets and we pick the k equal to 10, then we're going to reach to the within cluster distance to 0, because each of these data points are going to be a cluster itself. But there's no much information we can get from them, because we are just we cannot see the relationship between data points. So we don't want to have a really large k, but we want to have we want to do as good separation as we can. So for example, in this graph, maybe 4 is the turning point, where you have a relatively big decrease of the within cluster distance, but you still preserve some of the relationship between data points. Yeah, you just try to do something like this and find the optimal k you can get. And now I want to talk about the hierarchical clustering, which is an alternative approach to k-means clustering. Except for hierarchical clustering doesn't need any pre-specified number of clusters, and it generates this tree-based representation of the observation called the dendrogram. So as we can see from here, this is the hierarchical clustering. So there are a lot of different methods you can pick for this each cluster. You can check online how these are different. This is going to be a whole lecture, so I'm not going to go deep into it. Yeah, but we can just show you guys briefly what this look like. So for example, we have this dendrogram, and if we want to have like let's say 4 clusters, we can just cut it from here. So we know like these are the in one cluster, and whereas these are in another cluster. You want to only have 2 clusters, we cut over here, and we can see yeah, there is well separated. So not only that, hierarchical clusters doesn't require a predefined cluster number. It can also show like within each of the clusters which 2 samples are closer together. Let's say for we know like all these data points are clustered in the same category, but we know these 2 are more similar than this one from this one. So that dendrograms gives us more information that we need, and it's more flexible for us to define the clusters. So now I'd like to talk about batch effect. So what is batch effect? So batch effect are those variables that's not ideal and should not be ideally should not be there. So for example, maybe you have 2 different people doing the sample preparation, and maybe you do the sequencing at different institute, or you prepare the sample one batch at the morning, another one at the afternoon. So all of these factors can be considered as batches, and sometimes sample from the same batch might cluster together, but there is actually no biological difference within between batches. It's just the other factors are taking actions and are showing this difference. So most of the time, batch effect is going to deviate our finding from the real biological relevant information. So we want to try our best to eliminate batch effects, so we can get a real difference between conditions. So here I'm showing an example. So first I imported this count matrix, which is raw count. And then I use the log to TPM. I normalize the data and get the log to TPM. And from here, I then plot a correlation heat map between these samples, and we can see we can just see from these 4 samples, we see that these are all mouse model. So we can see mouse model from different tissue are actually showing a higher correlation than the mouse model maybe from heart, than human model from heart. Like they're very mouse heart. Yeah, so it's here. So they are much different than the is this one? Yes, this one. So they're much different from those between the so the correlation between the human sample from heart and mouse sample from heart is very different from mouse sample from heart and mouse sample from kidney. So the difference between mouse and human is actually larger than the difference between different tissue samples. So this is not as expected. And then to see this even more clearly, we can just use the PCA and do the scatter plot on the first 2 PCs, and we can see there is a clear separation between human and mouse. But this separation is not as expected, because we would expect to see human and mouse have very similar is very similar to each other, so we can use the mouse model to study human disease. If mouse and human is so different, we cannot transfer any of the knowledge we learn from mouse model to human settings. So this is definitely not as expected. And also, if we look deeper into this data, we can see the mouse model has a cluster 5, batch 5 and 1, and the human samples is from batch 3 and 2. So they're from different batches, and so we will not be able to say whether this is separation is because of batch effects or the separation is because of some real difference. So to eliminate this batch effect, we're using combat. So here we can see we in this function we pass in the log2 TP, the normalized data count, and the variable that we are interested in removal, so the batch. And after the batch effect, we do a normalization, we do a PCA on it, and we see the plot. So after the batch removal, we can see there is no very clear separation between human and mouse anymore. This makes so that tells us the first finding of this human mouse separation is actually not real, but from the batch effect. So now we're ready for DSIG. So the DSIG for DSIG we need to input the value in the matrix should be unnormalized counts or estimated counts for sequencing data or for fragments. It's important to provide count matrix as input for DSIG2 statistical model to hold, as only the count values allow accessing the measurement precision correctly. So DSIG2 model internally correct for library size. So transformed or normalized values such as count scaled by library size should not be used. And the DSIG data size object will must have an associated design formula. So the design formula expresses the variable which will be which will be used in modeling. The formula should be a tilde followed by the variable with plus signs between them. So for example, we can see it like right here. However, it's here I'm only using condition, but if you have other variables, you can also add a plus signs to separate these two variables. For example, if we have batch tilde batch plus condition, the design will indicate how the model how to model the samples, and we want to, and if the DSIG dataset is going to measure the effect of the condition controlling for the sample for the batch differences. So this function only this function is going to show you the effect of the last variable, and the previous variables that you put in is just going to be controlling factors. So firstly, yeah, let's start from back to here. I'm just importing some of this data, and we can see here there are two in the condition column. There are two conditions, which is the A and the B. And then I prepare for the DSIG dataset. And we want to see the effect of the condition, and then I do the DSIG. And the result table will be generated using the function results, which extracts result table with log2 fold change, p-value, and adjusted p-value. With no additional argument to result, the log2 fold change and p-value will be for the last variable of the design formula. And if this is a factor, the comparison will be the last level of the variable over the reference level. So in our case, the B versus A, which is because A is at the front and B is later. So B versus A, as saying here. Yeah, and if this is batch plus condition, that will be condition effect with batch controlling. If that's condition plus batch, then it's batch effect conditional on controlling on condition. So yeah, it's really important to figure out like to the order passing here is really important. And then we do a log4 change shrink for visualization and ranking. So the shrinkage of the effect size is useful for visualization and ranking of genes. To shrink the log4 change, we pass the DDS object to the function LFC shrink, and we specify to use the Apoglim method for effect size shrinkage. So there are different methods, but I think this one is the most commonly used one. I don't really know so much about the different methods. If you guys are interested, you can just Google them yourself. I have been using this myself, so I don't really know there's other stuff. And then if you want to show like how this is distributed, we can do this plot MA, and we can see there are some, we can see the log4 change, some of the genes has log4 change greater than 1. Yeah, and this can provide us this information. Sometimes you can even label, you can also adjust for the axis. So maybe you can see here there are some of the genes maybe further away, as more than log4 change 3. You can change this to -5 to 5, so you can see the whole picture. And sometimes you can even label the gene names of these genes that's has really different log4 change. You can also do a volcano plot, which is like this one, and this is called the enhanced volcano. So it's a different one from the previous year that I was using. That you need to specify all these factors to see which one you want to have the label to. But this one is much easier. You just need to pass in some of these values, which is not majorly just the peak cutoff and the log4 change cutoff are the ones that you're mostly interested, the thing that you want to specify. And if you want to, you have a set of genes that you're specifically interested in, you can just plot, like here, plot all these genes that you're interested to see where these genes are located in this volcano plot. So the volcano plot showing like the p-value and log2 fold change that passes the cutoff you have, and those that's only passing for the p-value is in blue here, and those that only passing the cutoff for the log4 change is showing in green, and those that's not significant is like gray. So this is even a better way than the standard volcano plot. Or if you're interested in the standard volcano plot, you can also take a look of it. Maybe you can you have some more freedom over that plot. Yeah. So after DSEQ2, we already have a list of genes that's overexpressed in either of these conditions. So now we want to look into the gene list and to see which of these biological pathways are these genes actually in. So there are two ways. The David takes the selected list of differentially expressed genes and sees if there are genes in the certain gene sets that are already expressed, where GC takes all the genes and use the test statistics. So for both of these, we need to have a good definition of the gene sets, and David has its own gene set definition. GC, we need to provide one by ourselves. So as MSIGDB will be a good one that's being commonly used to provide to GC. So we're going to do a very small tutorial on the David. So first, you already have a list of genes that's differentially expressed, like here, and then you click on choose file and this list of genes here, and then you select the identifier, which is this original one. You don't need to do anything on it, and you click on gene set and you submit the list. You submit the list and then you put Homo sapien, which is this, so 9606 Homo sapien, and you submit to the conversion tool. It's going to take some time for conversion. This is the one that I already converted, so I just select this converted gene list and select the specimen, and we do the functional annotation, and this is the result we get from it. So basically, after this annotation, you can after this cluster annotation, you can just download this file. You can analyze on other platforms. So GC is another way to do this gene list analysis. So in GC analysis, you need to ranking all the genes in the dataset, and you're identifying the rank position of all members of the gene set in the ranked datasets. Calculating the enrichment scores that represents the difference between the observed ranking and which will be expected, assuming a random rank distribution. So first, you already have the differentially expressed gene list, and then you need to take the rank. You need to take only the you can use this as a criteria, or you can use other things. That's a modification of this, but here we're using the log4 change. You take only log4 change as the rank, and you give the rank the same name as the gene list you got, and you sort the rank from positive to negative. So the positive genes are those that has like was differentially expressed in the first phenotype, and the negative genes are the genes that's that's overexpressed in the phenotype 2, just second phenotype. And then you're going to import some load some gene list as saying here that you will require not here, wait, oh yeah, right here, that you'll need to have a good definition for gene sets. So we're going to provide you with a PEGG gene sets, so you can just load it and pass it in here, and you have your ranks here, and you run the FGC, and you'll get something like this. So this is like so from this we can see the hallmarks oxidative phosphorylation has a enrichment score of 0.5, which is positive, so it's enriched in the first phenotype. And if we look, if we plot it out, we can see something like this. So this this top point is actually the enrichment score, and the leading edge is a subset of gene sets. That's a subset of members that contribute most to the enrichment score. For the positive enrichment score, the leading edge subset is the set of member that appears in the prior to the peak score. Or for a negative enrichment score, it is a set of member that appears subsequent to the peak score. And the bottom portion of the plot shows the value of the ranking matrix as you move down the list of the ranked genes. The ranking matrix measures a gene's correlation with the phenotype. The value of the ranking metric goes from positive to negative as you move down the rank list. A positive value indicates correlation with the first phenotype, and the negative value indicates the correlation with the second phenotype. Sometimes you're going to have a continuous phenotype. So the positive value indicates correlation, and the negative indicates no correlation. But here we only have two phenotypes, so it's not continuous. It's just showing either it's enriched in the first phenotype or the second phenotype. So, and this GC is not required for undergrads. But if you guys are interested, you can just Google online. There is a lot of tutorials on this. I think it's really useful, and I think you guys should take some time to look into it. Yeah, so basically this is it for today's material. If you have any question, feel free to ask me.